Dear children, as we discussed in the previous session about the reproduction in various organisms, at the end of the chapter, you must remember these terms. The questions can be framed from any of the terms directly or indirectly. Juvenile phase or vegetative phase, the phase when the organism is growing. It is not reproducing, it is only growing. That is known as juvenile phase. It is the first phase of different organisms. After that phase is over, the second phase is reproductive phase, the phase during which the organism is capable of reproducing and having a new progeny. After this phase is over, the last phase is senescence, that is the aging phase. That varies from different organisms. In some of them, the organism dies after this, and in some other organisms like the perennial plants, the organism shed its leaves, the plants shed their leaves, and then they are able to produce new leaves starting as a juvenile phase in them. Then what is lifespan? Since the time of birth till the death of the organism, the span that the organism spends is known as lifespan. Then what are the various reproductive units which are asexual units? Conidia, zoospores, aplanospores. These are the structures formed in lower forms like fungi, gemmules in sponges, and then in case of plants we discuss bulbs, corm, rhizome, runner, stolon, offset, etc. They are all acting as the mode of propagation in which the gamete cells and their fusion is not required. Then what are seasonal breeders? Those organisms that are capable of breeding only in a particular period of time, they are known as seasonal breeders. These breeders have a special mode of reproduction, and the animals are capable of doing and producing their eggs and male gametes or sperms only in a particular season. That applies to both animals and plants. Then meiocytes. These are the specialized cells that are present in an organism which are capable of undergoing meiosis, and these are usually leading to the formation of gametes in diploid organisms. Then in case of the organisms that are mammals, the reproductive cycle which is shown by the females is menstrual cycle in primates and estrous cycle in non-primates. Estrous cycle, these are the cyclic changes which a female shows during its reproductive phase, and during this cycle it is capable of producing a gamete, a female gamete, which can be used for a formation of a zygote. Then pollination. What is pollination? Pollination is the transfer of gametes from one anther to the stigma of the flower, which can be same flower or another flower. This is the part of pre-fertilization event. Second part of pre-fertilization event is gametogenesis, that is the formation of gametes, which can be oogenesis and spermatogenesis for female and male in case of animals, and megasporogenesis and microsporogenesis in case of plants. These are producing gametes. Pollination is leading to the transfer of gametes. These two are pre-fertilization events. Then next is fertilization. When the fusion of male and female gametes take place, we call it as fertilization, and the process is known as syngamy. After the fertilization is over, there will be formation of a zygote, which is the post-fertilization event, and this zygote will divide to give rise to an embryo. The process is known as embryogenesis. During this process, the cells will divide and they will differentiate to form different parts and different organs of the organism, both in case of plants and animals. Then on the basis of how the organisms are reproducing, they can be divided into oviparous, viviparous, and there is another term called as ovoviviparous. Oviparous are egg-laying animals, viviparous are animals which give rise to young ones, and ovoviviparous are a few organisms only where the eggs are formed, but what is a animal, a live animal. So they are called as ovoviviparous. Now in case of plants, after the fertilization is over, we discussed that the ovary will develop into a fruit, and inside the ovule will form a seed. The seed will give rise to a new organism or new plant, and it will be enclosed by the ovary, which is having different coverings together called as pericarp. This was discussed in class 11 also, that it is divided as epicarp, mesocarp and endocarp. So these are the terms that you must know in this chapter. But there are a few examples, like some plants reproduce only once in their life, like bamboo. And there is another plant called Strobilanthus kunthiana, which flowers once in 12 years. The last flowering was in 2018. Now you must all know and calculate that when should it flower the next time after 12 years. We discussed another term yesterday, that was the plants which are having life cycle of one season called as annual plants. Those having life cycle of 2 seasons is one-year plant, and the ones which keep on repeating itself year after year are called as perennials. So these are the terms that you must know after the completion of this chapter. The questions can be direct or indirect, and the examples can also be asked. Now since we have discussed in this chapter that how the different organisms reproduce, we are going to now discuss about how the reproduction occurs in case of flowering plants. So in case of flowering plants, reproduction in flowering plants, how does the process take place, leading to the formation of a new individual? Reproduction in flowering plants. Now in case of a flowering plant, that is angiosperms, the reproductive unit is a flower. And a complete flower, it can be a complete or an incomplete flower. A complete flower which is having both pistil and stem. Incomplete flower can be a flower which is having either pistil called as pistillate or stamen called as staminate. These terms we discussed in earlier also, that the flower which is having only female part is known as pistillate and the one which is having only anther is known as a staminate. So these are unisexual flowers having female and male flower separately. That is why the terms were used as unisexual, bisexual, monoecious and dioecious. So these terms are being repeated in the chapter, that a complete flower which is having calyx, corolla, pistil and stamen. All 4 bulbs are there. So it is a complete. This is not having all the 4 bulbs. It can be missing one of the bulbs, then it is known as an incomplete flower. So those plants which produce unisexual flowers, or those plants which are having unisexual flowers and male and female flowers growing separately on the same plant, flowers will be called as pistillate or staminate flowers. And the plant will be called as unisexual or bisexual depending upon whether same plant is having both the sexual organs or they are having different organs. You must remember the examples for both monoecious and dioecious plants. Now when we talk about flower, let us start with the reproductive units that are present in a flower and that are leading to the formation of male gametes. They are called as pollen grains in higher forms. Since we are referring to flowering plants, they are called as pollens, and the process of their formation is known as microsporogenesis. So when we talk about the main part, it is the stamen, which is having 2 parts, filament and anther. Filament is attaching the plant the stamen to the, or it is attaching the male part to the flower. But anther is the structure where the process of microsporogenesis take place, which leads to the formation of the male gametes or pollen grains. Now, anther is usually a structure which varies from species to species. That can easily be observed if you observe different types of flowers around you and you observe these anthers and their pollens on the slide. You will find that they vary from one plant to another plant. But the process of their development and formation is just the same. So, what happens here is that in case of these plants, the anthers are attached to the plant in a very different manner. Some are totally arranged and some are arranged partially. So that attachment varies from plant to plant, but when we talk about their development, the development seems to be similar in all the plants. So we are going to discuss about the process of microsporogenesis, the structure of anther, how it is formed and how it is attached. The anther is said to be diathecous and it is having the protective covering so that the pollen grains can germinate inside in a very proper manner. So that is what we are going to discuss now. Now when we talk about the main part, this is the stamen consisting of two parts, filament and anther. Now you can see that filaments and anthers, they are there and the whole structure is forming the bulb called endoecium. Endoecium contains stamens and the distribution, number and arrangement of stamens varies from flower to flower, species to species. But the basic structure remains the same, that they consist of filament and anther. Now anther is the main unit where the process of microsporogenesis will take place, and this microsporogenesis process will lead to the formation of pollen grains, which are the male gametes. Now we will discuss about the structure of anther and how the formation of or the process of formation of pollen grains takes place here.
Though the number and size of stamens in an androecium differ across species of flowers, the basic structure remains the same.
Now you can see the arrangement is varying from flower to flower, but they all are having one stamen. All stamens are having one anther and a filament, which can be of small size or a long size, which can attach either to the thalamus or to the petals.
Every stamen, for instance, consists of a filament and an anther. The filament is a long and slender stalk whose proximal end is attached either to the thalamus or the petal of the flower. The anther, on the other hand,
this is the structure of anther. Now, the anther varies in shape and size, but the basic structure of all anthers is similar. Like you can see that there are two lobes present in the anther and there is this longitudinal structure called a fissure, which is also known as the line of adhesion, from where the anther splits at the time of release of pollen grains.
With each lobe being diathecous or consisting of two thecas that are separated by a longitudinal groove measure.
Now, two thecas, that is two layers, they are separated by a longitudinal groove or a line of adhesion.
Runs through the anther.
Now when we have a look at the transverse section,
reveals that its lobes form a four-sided or a tetragonal structure.
Now, you can see that the anther in the transverse section has four sides and it is called as a tetragonal structure.
in the corner of each limb.
Now, if we take the enlarged view, a sectional enlarged view of the section of the anther, we will find various layers that are protecting and a specialized region where the formation of pollen grains will take place.
Is the microsporangium, a sac-like structure which is nearly circular in outline.
Now, you can see that all the four sides are having small circular structures called microsporangium. Now, these are the structures where the presence of meiocytes is there from the sporogenous tissue, and they will undergo meiosis to form the pollen grains, or they will undergo microsporogenesis to form the pollen grains. Now, these are forming various layers also.
Microsporangium is surrounded by a multi-layered wall consisting of the epidermis, endothelium, middle layers, and the tapetum.
Now you can see that there is presence of different layers that are protecting the microsporangium. The outermost layer is epidermis, inner to epidermis is endothelium, followed by 2 to 3 layers of middle layer called as middle layers also, and the inner one is known as, the innermost is known as tapetum. Now tapetum is a very important layer of cells which is having dense cytoplasm and can have more than one nucleus also. These are very important because these are the cells that are going to provide nourishment for the formation of the sporogenous tissue and the developing pollen grains. So it is a very important layer of cells here. Now, this is sporogenous tissue or the group of meiocytes that will undergo meiosis.
The epidermis, endothelium, and middle layers protect the microsporangium as well as aid in dehiscence, which
now you see that all 4 layers are protecting, and when there is fission in the longitudinal section, they also help in the splitting, in the dispersal and the release of pollen from the end. But the tapetum cells are helping in the process of providing nourishment.
the release of pollen grains. While the tapetum
This is also a very important structure. The previous one and both are important from both point of view. It can be asked either to draw or to label, and after labeling give the function of various parts. So both the diagrams are to be done. You can be asked to draw it or can be asked to label it and give the functions of various layers that we discussed just now.
whose binucleate cells possess dense cytoplasm.
Now, this is the innermost layer which I just told you that it is tapetum, which can have more than one nucleus also, and it is having a dense cytoplasm because their function is to provide nourishment to the sporogenous tissue and the developing pollen grains.
Nourishes the developing pollen grains. The sporogenous tissue, on the other hand, consists of a group of compactly arranged homogeneous diploid cells.
Now this is very important. What is sporogenous tissue? It's a tissue which is a mass of densely arranged cells which are also called as meiocytes, and it is these cells which are diploid in nature and they will undergo meiosis. Each cell forming 4 cells by the process of microsporogenesis which will develop into pollen grains. So it is very important that what functions are performed by various layers of enzymes.
Called microspore mother cells or pollen mother cells.
Now, why are they called as microspore mother cells? Because these cells are going to form microspores which will develop into a pollen grain. So also called as pollen mother cells or microspore mother cells or MMCs. They are all diploid.
These microspore mother cells divide meiotically to form haploid microspores.
In class 11 we discussed that meiosis consists of 2 stages, meiosis 1 and meiosis 2. Meiosis 1 leads to reduction of number of chromosomes and meiosis 2 leads to the equation of it. So each cell, each microspore mother cell is going to give rise to 4 microspores. So one microspore mother cell forming 4 microspores and which will ultimately develop into 4 pollen grains. This process of
which
formation
arranges
of
cells in a tetrad.
this process of formation of microspores from the microspore mother cell is known as microsporogenesis. The process of formation of microspores meiotically from a microspore mother cell is known as microsporogenesis.
Changes. While the microsporangium develops into the pollen sacs that extend longitudinally through the length of the anther.
Now you can see as the anther is growing, what happens? There will be fusion of the division ball and you can see only two pollen cells because the longitudinal section has now fused together to give rise to only two cells. Now this is one microspore mother cell giving rise to four microspores. Now each of these microspores will now develop into pollen grain.
The microspores separate from the tetrad and develop into pollen grains.
Now let's have a look at how this microspore develops into a pollen grain. Each microspore is a haploid. It will give rise to two cells of different sizes. And the process will be mitosis. So both the nuclei will be haploid. The smaller cell is known as generative cell and the large one is known as vegetative cell. Now you see, microspore is having only one nuclei, but as it develops into pollen, the pollen is having two cells, one small generative cell and the other large vegetative cell. As this cell is dividing and giving rise to the gametes, what happens? There is development of an additional layer outside the pollen grain, which is known as enzyme. Inner to that is a layer intern, and then is present the generative nuclei, vegetative nuclei, vegetative cell, generative cell. And enzyme is made up of a very resistive material called sporopollenin. It is so hard that we have been able to extract pollen grains that were present even millions of years ago. Now, since this element or this compound is so resistive that nothing can break it, so for the germination of pollen tube, there is presence of an area which does not have the covering of sporopollenin, and this is known as germ pore. It is through this structure, this pore, that there will be formation of pollen tube, because sporopollenin will not allow, it is so resistive that it will not allow the formation of pollen tube. So the areas where there is no sporopollenin present, they are called as germ pores, from where there will be formation of the pollen tube. The pollens can be shed either at this stage, this is known as two-cell stage, but this is not a functional nuclei, and now these two are known as male gametes. They are all haploid. This is vegetative cell, this is vegetative nuclei, this is enzyme, this is intine, germ pore, and two male gametes. It is this stage and this pollen which is functional Because in flowering plants we have already discussed that there is triple fusion. So triple fusion is possible only when there is presence of two male gametes, one leading to syngamy and the second leading to triple fusion, together called as double fertilization. So to ensure double fertilization, there has to be presence of two haploid male gametes, and it is at this stage that the pollen grain is capable of fertilization. So this is the structure and development of pollen grain from a microscope. So what we have discussed? We have discussed that anthericide structure where the formation of pollen grains takes place. The process is known as microsporogenesis. The sacs present inside are known as microsporangium. Inside are present the sporogenous tissue, which is consisting of compactly arranged cells. They are also myocytes, and they are capable of undergoing meiosis. The meiosis leads to formation of 4 microspores. One microspore mother cell will lead to formation of 4 microspores. Each microspore will undergo mitosis, forming 2 cells, one large vegetative cell, one small generative cell. As this process is going on, there is secretion of an external protein called enzyme, which is made up of a very resistive material called as sporopollenin. Since it is very resistive, there is presence of areas on the pollen grain where there is no sporopollenin, and that area is known as germ pore, from where the pollen tubule arise. Now, the pollens can be shed at this stage when it is 2 cells and 2 nucleated stage. But the functional stage is 2 cell 3 nuclei stage, in which the generative nuclei will divide mitotically and having 2 nuclei in a single generative cell, and these 2 nuclei are known as 2 male gametes. Why 2 male gametes are required? Because in case of angiosperm, we have studied that there is double fertilization, means 2 times fertilization occurs. One is with the egg cell called as syngamy, and the second is with the polar nuclei called as triple fusion, to ensure that there is presence of 2 male gametes. This is the process of formation of microspore and the formation of pollen grains.
Thus, the anther contains thousands of pollen grains which undergo mitosis to form 2 unequal cells. The larger cell, known as the vegetative cell, is characterized by a large irregular shaped nucleus, vacuoles, and abundant food reserves.
Vegetative cell is the cell which will help in the formation of pollen grain. That is why it is dense, large size, and it will provide initial nourishment for the formation of pollen grain.
While the smaller cell, known as the generative cell, is distinctly spindle-shaped and possesses dense cytoplasm.
Now, this cell, you can see that it is spindle shaped. You must remember for a 1 marker what is the shape of generative cell. Which cell is having the irregular nucleus? What is the function of vegetative cell? What is the function of generative cell?
Moreover, the generative cell floats in the cytoplasm of the vegetative cell.
This is very important. Which cell floats in which other cell?
Did you know that the vegetative cell and generative cell represent the 2-cell stage of the pollen grain?
Now there is one very important point, that in case of at the time of division of anther, what happens? The outermost layer, if they are thinner, they will break open, burst open through the fissure, and there will be release of the pollen grains. Now one very important point here is
the line of basins and result.
You can see the line of basins, there is splitting and the pollens are being released, and you can see that the 4 chapters have now become 2 pollen sacs.
The pollen grains are shed at the 3-cell stage whereas in 60% of angiosperms, which include plants such as hibiscus, the pollen grains are shed at a two-cell stage.
As discussed, the pollens are shed either at two-cell stage or two-cell three-nucleated stage, but for the fertilization they must have two male gametes and one vegetative cell.
Pollen grains, which represent the male gametophyte, come in a wide variety of sizes.
Now here you can see pollens are identification mark of the flower. There are different shapes, sizes of the pollen grains, so they are unique to the plant.
Shapes and colors. A typical pollen grain, however, is spherical in shape and has a diameter measuring about 25 to 50 micrometers. It is surrounded by two layers of wall, exine and intine.
This we have already discussed.
Exine, the hard outer layer, is made up of sporopollenin.
This also has been discussed that they are made up of sporopollenin, which is very resistive, and to allow the formation of the germ tube and the pollen tube, there is presence of germ core. Right? So now
they germinate into a pollen tube that carries the male gametes towards the embryo sac.
the vegetative cell initially gives rise to pollen tube, and then the male gametes follow, and they reach the embryo cell where the process of fertilization takes place.
This ability of the pollen grain to deliver the male gametes to the embryo sac is called pollen viability.
All pollens are not viable, all pollens cannot fertilize. So, if the pollen is capable of fertilizing, it is called as pollen viability.
However, factors such as temperature and humidity affect pollen viability.
You must remember what factors are responsible, what is pollen viability, what factors are responsible, and what is the tentative viability of a particular pollen. It can be 30 minutes, it can be 1 day, it can be a week or a year also.
Moreover, pollen grains of different plants have different periods of viability. Pollen grains of cereals such as wheat and rice lose their viability within 30 minutes of their release.
This is very important, that why all pollen grains are not capable of fertilizing, you must know, because they lose their viability. Especially in case of wheat and rice, the pollen grain is losing its viability within 30 minutes. So if the pollen is reaching the stigma after 30 minutes or in 40-50 minutes, though it is showing pollination, but pollination does not ensure fertilization. So this is very important question, that why all pollens are not able to fertilize.
Whereas pollen grains of some members of Rosaceae, Leguminosae and Solanaceae remain viable for months.
Now, these are another group. I told you that they can be viable for a few minutes, weeks, days or even a year. So these examples you must remember.
Pollen grains, which play a vital role in plant reproduction, are also often consumed by human beings as they are rich in nutrients.
Now, pollens are important not only for plants. They are now being used as a source of energy by human beings, and they are available in the form of tablets or syrups that are being used to increase and enhance the activity.
Pollen products in the form of tablets and syrups are commonly spotted on shelves of supermarkets. Moreover, pollen is also stored in pollen banks in liquid nitrogen and water.
This is very important. Why do we have to conserve the pollen? Pollen needs to be conserved because at the rate which we are losing the various species, it is mandatory for us to conserve certain viable pollens so that they can be used in future. And this is done in liquid nitrogen, and the process is known as cryopreservation. It is at minus 196 degrees Celsius, liquid nitrogen. The pollens remain viable, and as and when desired, they can be brought back to normal temperature and can be used for process of fertilization.
196 degrees centigrade. The pollen is later used in plant breeding programs. Pollen grains, however,
So, pollens are conserved in the pollen grains by the process of cryopreservation, so that can be used in future also. But some pollens are having a negative impact also, and they can trigger different kind of allergies, mostly in case of asthma patients.
can trigger allergies in some people and can cause asthma and bronchitis. In fact, the reed carrot grass or Parthenium hysterophorus and its pollen, found in now cultivated lands in Punjab, causes allergies such as eczema, dermatitis, and other skin diseases.
Now you must remember about carrot grass or Parthenium, which is one of the major cause of allergy in certain areas where they grow.
Pollen, formed inside the stamen, plays an important role in plant propagation.
So, pollen, the way they mix, they are the pollen grain, the viability is important, their formation is important, and they are otherwise also used by humans to enhance their activity because they are rich source of minerals and other substances. With this, we wind up the topic, the development of male gametophyte or the process of microsporogenesis and the formation of pollen grains. Thank you.